Hi, everyone. Good morning. Welcome to the Delegate Assembly today. Hello. So as we get started, I've put the link just to the little initial slide deck in the chat. I'm going to copy and paste it in for everyone who's still joining, just in case you want access to these slides. So before we get going with the actual tutorial, and today we're doing image display. Right, so as you're joining, go ahead and log in at data.lsst.cloud and choose the notebook aspect. All of our tutorials today will be in the notebook aspect. I'll put that in the chat too. Data.lsst.cloud, that's the Rubin Science platform. Choose notebook aspect, get yourself logged in, and then you're going to want to be open at least notebook 03A, and that's in your notebooks / tutorial notebooks directory 03A, blah, 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 .ipynb. Put that in the chat as well. And also as a reminder, if you were with us for data preview 0.1, so you're a returning delegate, your notebooks / tutorial notebooks directory should have updated with all of the newest versions of our tutorials when we updated them. But in some cases it might not. You might have like a issue with Git in that in that directory. So as you open up notebook 3 for today, the easiest way to check if you have the most updated version of notebook 3 is if the last verified to run date is 2022-06-27. So you can check that, and if it does say that, you know you have the most up-to-date version of the notebook, and it'll be the exact same version that we're going to be using today. So make that check. If you don't have it, then you have to take some action to get the most recent version. If you're a Git user and you know how to use Git and you sort of understand things like Git reset, then you can follow the directions that we have at this link here, which I see more people joining. Stop it. See more people joining. Just trying to copy paste the link to these slides, and it keeps opening the slides. Okay, so if you just joined us, now you have the link to the slide deck in the chat as well. Oh, come on. Another temporary fix, and this is if you maybe know a little bit about Git, to get just all of the latest versions of the notebooks for yourself in a different directory, you can use these instructions to just git clone a whole copy of the repo for yourself. That's a pretty quick solution as well. But if you really don't know anything about Git yet and you just want to grab the individual copies of the notebooks that we're going to use today, this is a fast temporary fix. Is you can navigate to github.org/Rubin-DP0. I'm putting this in the chat. Go to the tutorial notebooks repository. I'll just show you actually. Okay, so if you need a copy of the notebook, you would come here, github.com/Rubin-DP0, go to the tutorial notebooks directory. 03A and 03B are the ones we're going to use today. So you can save the copy of the notebook as, and you would download it, and then back in the Rubin Science platform, if you wanted to upload it, say to your home directory, there's a button here, it's a little up arrow, and you could upload that file that you just downloaded. So if you do not have the most recent versions of today's notebooks, then that's how you would get them. So I'm going to give you some time to make sure you can open notebook 03A. I don't see anyone posting in the chat that they're having issues or that they have any questions about where to find today's notebook. Okay, if you're already in the Rubin Science platform, you go to your notebooks directory, tutorial notebooks, it'll be right there for you. And it should look like mine looks. I'm just pausing for a second just to see if anyone is having trouble logging in, opening the right notebook. Okay. And then the other announcement for today, I just wanted to preview what's coming up in the delegate assemblies. So today, we're doing visualizing image data, and then we're going to have breakout sessions in the second hour. 2 weeks from now, we will have a tutorial that's pretty dedicated to use of the Butler. So we'll be doing data query and retrieval with the Butler in the first hour, a beginner level notebook. Okay. Nandini will get someone to help you. What kind of trouble with O3A? Can't find it, can't open it.
So it says file load error for O3A.
Yeah, that sounds like a corrupted file. So your best and fastest bet is to go and just download a new version of it.
Okay, thanks, Melissa. Yeah. Sorry.
No, that's fine. Yeah, that's that error, that corrupted file. That's usually it's been
yeah,
edited.
I've had it also before with the image analysis tutorial, but I figured I was doing something wrong. I will change it now. Thanks.
Yeah, no, it's not you. And I think like eventually you'll want to go and take this action to refresh your entire, refresh your whole directory. But for now, just grabbing a copy of O3A and uploading it is going to be fastest. Yeah. Right, so 2 weeks from now, we'll focus on the Butler. And then I just wanted to pop up what's going to happen in the fall. We will continue with delegate assemblies, but Friday, September 2, there won't be one. That's a long weekend. So it's the Friday of a long weekend. So technically Monday is the holiday, but we're just going to give that a skip. Everyone have a nice long weekend. And then we'll pick things up again in the fall on Friday, September 16th, and then Friday the 30th. And we're going back to our more normal delegate assembly setup where we always have a tutorial in the first hour and breakouts in the second hour. And we're going to be doing source detection and measurement and then big data set visualization in September. And then later through the fall, there'll be lots of time for delegate presentations. So if you've been working on something, I know a bunch of you have been working on stuff and have already sort of suggested that you might present. So I'm really looking forward to those. We'll get that schedule set up soon. All right. Greg, your hand is up.
Yes, I just wanted to mention that those of you who participate in Stack Club, we're not going to have an instance of Stack Club a week from today because many of us will be at Projects Community Workshop in Tucson, and we'll skip that this coming Friday. That's all.
Thank you, Greg. That's a good reminder. No Stack Club next Friday. Yes. Okay, let's get started with today's tutorial. Image display and manipulation. So in this notebook, we're going to learn how to retrieve images from the Butler and display them using a package called AFW display, which is part of the LSST science pipelines. So as always, if you're new to notebooks, I'm going to try and describe like the components of the notebook as we go as well, just for added reinforcement. So right up at the top, we import our Python packages, and you can see we divide them up here just to give you a little bit of extra information. Some general Python packages. AstroBi is a common package used for astronomy, and then we have all of our packages that we import from the LSST science pipelines here. In this particular notebook, we define, I think there's 3 functions overall that are defined, and they're both sort of like shortcuts, so we don't have to copy paste these blocks of code over and over. So they're handy little functions that are defined right up the top that get used later on. So they get called later. One of them is to remove figure, and this is just done to save space. So every time we make a figure, we then remove it so that we're not keeping all of that data that we just plotted in memory as we move through the notebook. The second one is a handy little script to make an image cutout of data set type of your choosing. In this case, it'll make as a default, it'll do a cutout of a deep coad at a given right ascension and declination. So you saw this doing this like a little bit like finding images based on RA and DEC in notebook number 1, but this kind of just puts all of that functionality into one handy little code snippet here, where you can just pass it the RA and DEC that you want, and it'll go use the Butler, which we cover in 2 weeks. It'll use the Butler to grab the image that you want all at once. So that's really handy, and that's something that you can just copy and paste and use in your own codes as well. You should always feel 100% free to take code that we have in our notebooks here and use it for yourself, use it in your own notebooks. That's what we want you to do. We want you to be set up to use these little modules. The third function that we define right up here at the top is one to create an RGB image. That's a color image. Red, green, and blue are what RGB stands for. And so this is something that we'll use later on to make a pretty color image down. I think it's in section 6 of this notebook. So as you're going, I've already executed all of the cells in this notebook, but as a reminder, you want to be executing the cells as you go. So with your cursor in a cell, you hold the shift button and press enter, and it'll execute that cell. So as you go, as we go through each of these, you should have done shift enter in this cell, and this one, and this one. And you can see it gets the little number next to it as you execute them. So do be executing these cells as you go. After we define those 3 functions that we're going to use later on in the notebook, well, this is just we set up a bunch of plotting parameters, and this is all for the matplotlib package. So these have been set up just to make our figures look pretty. And this is, again, another thing, these parameters are something you can copy paste into your own notebooks if you like the way these plots look. All right, so shift enter in this cell as well to execute it. And we move on to section 2. Now we're ready to load some data to visualize. Right. So in this first cell here, we're using the Butler, which again, we will talk a lot about 2 weeks from now, but for this particular notebook, we want to focus on image display and image visualization and just use the Butler. So this is sort of getting your feet wet a little bit with the Butler and just using it without, having to know everything about it, which is fine. So we set up the configuration and the collection for the Butler, and then we instantiate it. So now we have a way to query the image catalog and retrieve images from where they're stored. The Butler works by defining what we call a data ID. And in this case, we define the visit number and the detector of the observation that we want to retrieve. And in this case, we're just starting with already knowing what the visit number and the detector are, but there are, of course, ways to find that out using the right ascension and declination and a date. You can find the visit number and detector. That's all 2 weeks from now, and how to use the Butler. We'll go into more detail into data IDs and that kind of thing. For now, we really just want to grab a image for you to play with. So we're grabbing this one, visit number 192350 from this specific detector. And we say, okay, now we use the butler.get statement to grab the calexp for that particular data ID. And a calexp is just the short term for the processed visit image. So this is not a stack, it's just a single image obtained at a single time in a single filter. And the visit uniquely identifies the filter as well, because it was a single image. And I don't think it says here what band this visit is in, but we're going to find out. Is someone taking notes today? Do we have
Yes.
Tina?
Yes,
yeah.
I am.
Yeah, can you make a note that we need to add what filter this is going to be?
Yes.
Yes, cool, okay. All right, so execute this cell and it'll Butler get, and it'll get that calexp and it'll store it as the word calexp here. And we'll learn more about the Butler next week. So shift execute to execute that cell, always shift execute to execute every cell as we go. And do shout, I can see the chat. So do shout in the chat if you encounter any errors. We have lots of Rubin staff on hand today to help you out as you go. Oh, and I will mention that it's an I-band image. Okay, cool. This particular visit is an I-band image. These errors are normal and they're going to go away pretty soon. So we just skip right on past them. Okay, so now we want to set up a little bit in advance to plot, to actually take a look at this image that we just retrieved from the Butler. This is just to set up matplotlib as the back-end default display for AFW display. And we are ready to display this calyx that we got, which is done in this cell here. So, oh yeah. Oh, I think, Simone, you might have missed the instructions right at the start. So,
yeah, I might. I connected 2 minutes. Yeah.
Yeah, so you do have to go to data.lsst.cloud.
Thank you. I'm sorry.
Yeah, no, it's fine. Log into notebook aspect. And
do you have some slides where I can find instructions so you don't waste your time?
I do. They don't. Yeah, there's the slides. Let me just. Once you're in the note. Yeah, once you're in the notebook aspect, you want to navigate to your tutorial notebooks directory and open up image 3A.
Okay.
I have a 3A notebook. Yeah, image display. Yeah.
Let's see if I can do it.
Yeah, and then that should get you. Yeah, nothing will execute from GitHub. You have to be logged into the Science platform.
Okay.
Yeah. Thank you, Douglas. Yeah, Douglas is going to help you out. Make sure you get set up. Okay, so to display the image, first we set up our figure, then we set display to be AFW display.display, where we want to display within that figure that we just made. We can set the scaling for our figure. You can see here too, the next step is to actually display that calyx that we retrieved from the Butler. And if you execute this cell, you will see that the mask has been displayed with it. Where a lot of the pixels are colored blue, and those are like, I think those are footprints maybe in the blue. We're going to talk about masks a little more, but my point is that there's a bit of a typo in this notebook. So actually, Tina, if you can make a note of that too, this should be calyx.image, because right now what we're displaying is like all the information that we got from the calyx, which includes all the mask values, where really for this first step, we just wanted to display for you just the image, like just the pixel values. So if you type .image there after the calyx, so we just want pixel data from the calyx, and we only want to plot that, and then shift enter to execute this cell again. Oh, you're tiny. Why are you tiny now? Something has changed. Oh, it's probably because I redid the imports. Well, that's okay. Just have a tiny little image, but you can see that the mask isn't plotted anymore. So now it's just pixel values being plotted, which is probably what you want for the first one. And I don't like that it's small. So I'm going to clear everything and just run up to here. Yours stayed the full size. Yeah, I think it might be because I re-executed the import cell twice. I think something weird happened. That's good that yours stayed full size. Thank you for that. So this, yes, this is what yours should look like as well. Okay, so you'll notice too that the scales here, like the X and the Y axis, are in pixels, not in RINDEC. And so we want to provide you with a really simple way to just put down an RINDEC grid over top of an image. And that's what the next cell does, where we have the same, start out the same way with defining our figure. And then we use the AstroPy WCS package, and we get the WCS from our calyx that we got from the Butler, and we pass the fits metadata to this WCS package from AstroPy, and we set it as the projection. That's going to let us put a grid down on top of our image that will be RINDEC. And then in this case, instead of using AFW display to display the pixel values, we're using Matplotlib's imshow. So this is just giving you another way to display images. So if we shift enter in this cell, you will see that it's the same pixel data, but now we have our index grid overlaid on top. So that's handy. In section 3.2, we go back to that mask plane that I told you about before, which comes with the CalX that you retrieve from the Butler. And so we can start by, in this cell, we're going to make 2 plots side by side of the image itself, which you'll recognize. So we set up 2 displays, and in display 1, we plot CalX.image, just like we did before. And then in display 2, it's going to plot the CalX.mask plane. So you'll see 2 side-by-side images. Of the image that we've already seen a bunch of times, our CalX with the mask plane over top. And then as an option here, instead, we're plotting just CalX masked image, which now you can see it's the image with the mask over top, which looks pretty similar to the first time when we plotted just the CalX without the dot image, where you see this have some shading, some like transparent shading over top of the pixel values that tell you about the mask. And so the mask can be pretty useful because it has a lot of extra information about the image. So in the next cell, is there only one mask that comes with the image? Does it mask saturated stuff? I believe, and I think in one of these cells down here, oh, because they're not executed for me anymore.
Yeah, I think once you get to showing all the mask bits, that'll be visible. I think that's just 2 cells from now.
Yeah, that's why I had it all executed in advance before. But yes, Louise, I think we're about to answer your question. So let's go ahead and execute this cell as well, where we're doing basically the same thing that we did above, but changing the mask transparency. And in this case, you can set. So there's various like reasons why you have a mask, which we're going to tell you about in a moment. So there's various like reasons for pixels to, have a mask. And in this case, if you want to mark all pixels that are detected as a source in your image, and you want to mark them as blue, that's what this is showing you how to do. So we're setting the transparency, and then we're setting mask. When the mask means it's detected, we're setting it to blue, and then displaying it again, and then you see it here in that case. And so all of the different reasons why pixels are masked come with their own preset color. And so, so this is where we can see that bad pixels will be marked as red, cosmic rays as magenta. When you're near the edge of an image, there can be artifacts and issues with that. They're marked as yellow, and so on. And so here, so to answer Louise's question, saturated, there is a mask for pixels that are saturated, then they would be green in the image. Yes, okay, yes, we're answering those questions, cool. So actually, Tina, can you make a note too that in this tutorial, we might want to move mask definitions above mask display? Because a lot of people, I think that sounds good. Right, and as with all things Python in the notebook, you can always type help, and then most of the time you can type help, the thing you want to learn more about, and it will output a bunch of information for you. So all of that info is there for you to keep learning as you go through these notebooks. And sort of on that theme of getting more help, you can do that as well for things like all the methods associated with display, how to use the display scale functionality. What is this MTV function that we use to display the images? All of those help files are there embedded, and you can access them from the notebook. Okay, so that was displaying, remember it was a CalX, so it was a single visit, an image from a single visit in a single filter. And now we want to show you how to extract a coad, so a chunk of the deep stacks that are made from all of the images, and display that instead. So here we're going to, we've again set the data ID for you, where tracked and patch, if you remember, tracked and patches are how the deep all sky coad is divided up. So we have the all sky deep coad of all of the, made from all of the images, divided up into tracks, and then those tracks are divided up into patches, where a patch is like a nice human visible, like human workable size image. So often we grab patches when we want to visualize deep coads. It's like about the same size as a CalX, I think. So we've set this one up for you. We've already pre-chosen a nice area of the deep coad for us to look at. So we again use butler.get to grab it. And then we use the same set of commands to display our coad. You can see here we're doing display.mtv coad.image. So we're just displaying the pixel values this time. And there we have it. It's a very nice image to look at and play around with. And you'll notice here that since we've patch is part of a larger tract, and so when we have the pixel values here along the sides, they aren't starting at 0, 0 like they do for the CalX, because this is, we've got a chunk, we've got a patch out of a larger tract. The coad has the WCS embedded in it, just like the CalX did, that you can retrieve. And if we say, oh, well, that really nice cluster looks like it's around pixel values of 12,000, 8,000, we can use that WCS from the coad to convert those pixels to sky coordinates to get the right ascension and declination of that really pretty looking galaxy cluster that we see there. And now that we know the RA and dec of that nice looking cluster, we can use our cutout underscore coad function that we defined up at the top. And this is exactly the same, documentation from that function that was defined up at the top. And we're going to use our cutout underscore coad function to make a coad cutout that's centered right on the RI and dec of that cluster. It doesn't say here what filter we are grabbing the coad for, but the default was R band. It should say the default here too, but it doesn't, but it was R band. So we're grabbing an R band cutout and using the exact same set of functions, set of commands to display it as we always have been with AFW display. So this one here, this before, and now we've got a nice like centered in view of this particular galaxy cluster with all of these beautiful galaxies there. Just going to pause for a moment. Make sure you've all had time to execute these cells and are keeping up. Okay, nobody's yelling. So we'll move on to overplotting catalog sources onto this coad that we've just pulled out. So, right, overplotting catalog sources. In the last tutorial 2 weeks ago, we covered the TAP service for catalog access and how to query the catalogs and the tables using TAP. But catalog data is also available via the Butler, and you can retrieve it using the same process that we've been using to retrieve images, which you can see here, we're using a butler.get statement. Oh, a question. I'll leave Jeff and Douglas to handle that question because I do not know the answer right off the top of my head. And that was, yeah, that is definitely not covered. I don't recognize that error. So retrieving table data from the Butler, we're using a butler.get statement, the same statement that we used to call image data from the Butler. But in this case, we're passing the data set type. Previously, we'd use deep coad because we wanted data set type deep coad, but now we want to get data from the object table instead. So we've set data set type to the object table, which, as a reminder, object table are all of the detections in the deep coad. So since we've retrieved a deep coad and now we want to overplot the sources that have been detected in that deep coad, the object table is the appropriate one to use. So we are getting objects from the object table from the Butler using the same data ID as we use to retrieve our deep coad. So that's very handy because you don't have to set up like a separate tap query. You've already got your data ID that you use to retrieve the image. You use the same one to retrieve catalog objects. So we execute this cell and it got us 37,000 objects from the catalog, from the objects table that overlap with the tracked and patch that we had previously retrieved for the deep coad. You can take a look at this table. So this is printing out a truncated table, right? There were 37,000 rows, but it's only going to show us just a little bit. And there are also hundreds of columns in this particular table. So if you sort of scroll left to right, you can see that it's truncated as well and left out a bunch of columns in the middle. But it's retrieved all of that data for us. And now we can, since we've got, well, it's definitely showing cord underscore deck, but there's also a cord underscore RA. So there are the RA and deck in this catalog, and also the X and Y, the pixel coordinates are also in there. And so what this next cell does is our cutout image, it has a property called get bbox, which is basically the bounding box of the particular cutout that we got. So we want to know what those boundaries are. And then in the next line, we want to select all of our coad objects that have an X and Y pixel values that are within that bounding box. And then we can say, okay, our new array cutout underscore object is a subset of all of the coad objects that we retrieved with that data ID that are just within the bounding box of our like much smaller cutouts. So we execute that cell to select just objects that are actually in our little cutout image. And then in the next cell, we do same as we have over and over, displaying the image. And then on top of that image, we're going to display a plus sign and a circle at the location of every object that we retrieved from the Butler. Yeah. And there you can see all of the object table entries overplotted on our deep coad. And so another thing to remember here is the object table uses all of the filters. So it's the sum of everything detected in coads for U, G, R, I, Z, and Y. But this is only an R band. So you can see that in some cases, there are objects in the object table that aren't detected in R. So we don't actually see them in our image, but they're in the table. So they get overplotted on here. Just gonna pause another second before we make a color image.
Sorry, Melissa, is it the R or the I? Because in the earlier catalog,
it says I here, but if we go back to the cutout coad, the default band is to get an R band image, and we never specified differently.
Right. Okay.
Yeah, so that cutout is in our band yet. So
And the catalog is from the eye.
nope, the catalog is from the object table, and so it's got all of it's got all bands in it.
Okay.
Yeah, let me just pop back to, so we got extract to coad cutout. Wait, is that the right spot? Ah, it's here. Yeah. Here we got the cutout. We didn't specify a band, which means it defaults to R. And then when we retrieve objects from the object table, the objects is the sum of everything detected in any of the deep coads from any filter. So there are columns for measurements. You can see here, there's columns for measurements of the U band, but there are columns for measurements for U, G, R, I, Z, and Y in the object catalog. So it's a sum total of all detections. And so it's
Thanks.
this part that should say R band. Yeah. Oh, caps is on. Okay. Yeah, the calyx was an I band, and then this one is R. Thanks, Tom. All right, let's make a color image. This is the last step in this particular notebook, and it's pretty fun. So in this case, we're using that cutout underscore coad function that we defined before to grab the GR and I band images of this particular RA and deck that we had previously defined. So we grab all three, and then this line here puts them into a list of coads here. And so now we're ready to make our RGB image, red, green, blue image. And for this, we use that function that we defined up at the top to create underscore RGB. And this is going to plot two images side by side. On the left, we leave everything at, like, we don't specify a scale. And so we leave it at the default scale for these images. And then on the right, we set a scale. So you'll see the colors are a bit different in these two. So we have our original when no scale setting is done, and then we have a rescaled version, which you can feel absolutely free to play around with. Change the scale for the red image, for example, lowering that scale for R. So I changed this one to 0.4. Nice mistake. This should be fast. There we go. So I lowered the scale for R from 0.6 to 0.4, and our image got less red and more green, and kind of looks very Wicked Witch of the West. Let's change, I want to change the center one to try and see if I can make something prettier. By now compensating for lowering the green scale down to 0.4. And now I have a pink image, and that's much nicer to my eye, and I'm much happier with that. So that's my pink galaxy cluster, and you should feel free to play around with that. And then if you want more information on AFW display, etc., we've got some additional documentation down here at the end. Yeah, exactly, Simona, it changes. And then, yeah, we have this remove figure that's automatically doing that for you. Oh, to close or erase table data as we go. I generally do delete table data as I go sometimes. And especially if you're making lots of calls to the Butler and you're, pulling out lots of data, it can be helpful to erase or delete data that you don't need anymore in a notebook as you go. It's a good point, Louise. Okay, I think at this point, I'm going to pass it over to Jeff. You ready to do 3B?
I sure am.
Great, okay. Let's leave AFW display for a while and use Firefly instead.
Let me see if I can. Okay. Okay, so hopefully you can see my screen share now.
Yes.
Excellent. And as, let's see, sorry, I can't see you all. Let me see if I can fix my window so I can see people and chat as well. Okay, I don't know where the chat went. There it is. All right. Okay, so yeah, so now we're going to see a different way of examining images, and this is going to be using the Firefly interface, which is an interactive interface. If you're familiar with DS9 or something like that, this will look similar to that, and we're going to mostly be using the same images that that we used in the previous notebook. So most of these data products are the same that we're using for this notebook, and we're just basically, if you recall, when we set the back end for plotting and we set it to matplotlib in the previous notebook, now we're going to set that to Firefly, and you'll see that in just a second here. Okay, so as always, we do some basic imports. We need a Butler to be able to grab data products, and we need the AFW display package to display images. And then this cell just suppresses some warning messages. It's not vital, but it just gets rid of some things shouting at you. Okay, so as we did before, we need to select an image and load that image from the Butler. So this is the exact same thing that we saw in the previous notebook that Melissa went through. So we're just selecting, and in fact, it's the same visit and the same detector. And we did note here, since this came up in the previous one, oh, I can't remember which band this was. That's because if you specify the visit explicitly, then the band is sort of implied. So you could specify here band I, but it's redundant because this visit was taken in the I band. So you don't have to explicitly specify that. So there's a note up here about that if you forget about that. Okay, and again, we get this error that's going to go away soon. And now we want to display our image just like we did before. And so, as I mentioned, the way we use Firefly rather than matplotlib is just by setting the default backend to Firefly here. And then we initialize a display and we say frame equals 1 here. And so you can open multiple frames by and access them, I should say, too, by setting different frame numbers. But we're going to do this. We're going to execute this. And a new window will pop up with Firefly. And so in case you've forgotten, you can always switch between the tabs that you have open in the RSP, but you can also reorganize them by doing something like, if I grab that tab and I do like this, so I've selected the bottom half now, then you can see that this is the notebook, and we have the Firefly browser down here. Likewise, I can place it side by side like this. And then to put it back, you just drag it over here onto the next to the other tabs. So we have a Firefly window open now. I'm going to leave it. I don't know. I can't decide. This makes it hard to read the notebook. Yeah, okay. I think this is okay. Okay, so we've opened Firefly, and now just as we did before, we're going to use this MTV method to display the image in Firefly. So when I execute that, voila, we have an image pop up over here. And you can see, as before, by default, it displays the image with the mask plane overlaid. So that's why you see the blue halos around everything here. You can, if you want to display it without the mask, you can just say calyx.image, and it'll just show the image plane without the mask. But we're going to go back to with the mask plane so we can show you other ways that you can interact with that. Okay, so with that image open now, we'll show you how you can manipulate some of the oh, good. Yeah, Melissa's on it. All right. You can manipulate, for example, how the mask displays, or how the image displays this sort of color scale and things like that. So for example, you can change the mask transparency. I think by default, it's semi-transparent. You can see objects through. I'll note if I zoom in, you can see that there's an object behind here, and you can see the mask on top of it. But you can, it's somewhat transparent. So if I change the transparency, it goes from 0 fully opaque to 100 fully transparent. So let's try setting it to 20 and see what happens. Okay, so now it's more opaque. You don't really see the objects behind. And then if you want to switch it back to 80%, for example, there we go. So you can do that from the notebook. And you can also control the scaling, the color scaling of the image. So this is just an example, and you can play around with this, changing the values. And here's what happens when you use this. It's an eighth-inch hyperbolic sine scaling. So you can see it changed the image. Here's another version where it's gonna, instead of explicitly setting the upper and lower bounds of the scale, you can use this Z-scale algorithm. Try that and see how it changes. Oh, Melissa, yeah, go ahead.
Yeah, looks like a bunch of people, the Firefly just didn't open, and then they tried restarting the kernel and rerunning, and it just doesn't open. And I was wondering if we still have Gregory, is there other troubleshooting that we could try to make their Firefly windows open?
It's not obvious if you're just seeing absolutely nothing happen at all and there are no error messages in the notebook and no tab appearing. I'm not sure that there's anything that you could do other than, try to, I suppose, exit your notebook session and come back to it and try again. I'm just starting to try to duplicate this myself to see what happens running this notebook. So if it happens for me, then we'll dig into be able to dig into it more easily. I wasn't quite clear from the messages that were posted whether it was a lot of people or just one person. As if there was another message from Louise that said it was only for one of us that it doesn't work, so not sure. Let me look at it a little bit offline, and then we'll see if a pattern emerges. I'll poke at it here.
Okay, thanks. Yeah. People are reporting mostly their Firefly tabs are opening. So I think maybe it was just one person today didn't have the tab open.
I mean, you know it's a tab inside the Jupyter Lab session, not a browser tab, right? The person who's reporting that it's that it's not opening. That it does not open a new browser tab. It's inside the Jupyter Lab interface.
Yeah.
Okay, great. Okay, just wanted to be sure. Okay. Yeah. So
Okay.
mine opened right away. So we will. So Louise, if that's reproducible, maybe later in the breakouts we could do a shared screen thing if you're comfortable with that, and we can see if there's anything else we can figure out.
And someone else reported that their Firefly tab maybe didn't open yesterday, but then they totally logged out and did a full refresh and came back to the RSB, and it worked again. So
there's also a question that might be useful just to demonstrate again how you combine how you combine the two tabs together in the same display. I know that was always a tricky when I first learned about that. It probably helps to have maybe to show it once again, if that's possible.
Yeah. And so for example, I'll show you the backwards version first, since we have side-by-side windows. You can just click and hold your mouse button on this tab and then drag it over here to the where you see all the other tabs. And if I drop it, now it just joined these other tabs that I can toggle between. And so then to go back to, say we want this one top and bottom, if I grab and drag, you can see the top half of the screen turns blue, or I can say the bottom half turns blue here. If I let go of that, now it shows the Firefly in the bottom half and the notebook in the upper half. And so I can rearrange it, and I'm going to put it back side by side by dragging it to the right side. And when I let go, it snaps into place. And you can do that, it doesn't have to be Firefly that you put over there. You can also, I can do that with a different notebook. So now I have this notebook and Firefly all open, but I'm gonna, oops. And then I have to figure out how to put it back. There we are. Yeah, so that can be very handy also if you're sort of using a different notebook as reference or something like that. You can have them open side by side or top and bottom so that you can refer back and forth between them.
Great, thanks, Jeff.
Yeah, it's a very handy thing to remember, because often we're working with limited screen real estate, so it's nice to use it as well as we can. Okay, so we've already had some, I think we've had some demo of Firefly previously, but you can, I just wanted to note that you can, while you have an image displayed, we showed you here how you can change things like the mask transparency or the color scaling and things like that. But you can also do that within Firefly itself. So you see this thing that looks like a painter's palette. If I mouse over it, it says color drop down, change the color table. So if I click on that,
that's the image color table, Jeff.
Yes. Yeah.
Yeah. Okay.
Yeah. Good point.
Yeah.
yeah, so that's to change the image. So I don't know if I like a blue plus rainbow image color table, I can change that. Let's go back to what we had originally. And then, yeah, and then you can also, the mask planes, which we'll speak a little more about next, but you see this, this looks like a stack here. I'm not sure what yeah, there we are. This is the overlays menu. And you can see when I clicked on that, this shows you all of the masks and what their colors are. And you can see there's a checkbox by each one. So for example, the interpolated pixels, if I don't want to show those, but I want to keep all of the others, I can toggle that. And you can see actually on this bleed trail from saturation of this bright star, you can see those pixels that were masked disappear. And so I'm guessing that the other one, if I take the saturated one away, that green artifact will disappear, which you can see here. So you can control those directly here or in the notebook as we showed you. And then the scaling of the image, that's this, this stretch drop down. And so if I open that, you can see I can change to a linear stretch instead of the async stretch. And we patiently wait, and there we go. We have a linear. And also, since I have a wheel on my mouse, so I can just zoom in and out by rolling the mouse wheel, and of course you can grab the image with your mouse and move around in the image. So we'll come back to that in just a second. Just to note, this is what we did in Melissa's demo as well. If you want to see what those mask colors are, so that you know how to interpret what these mean, why does everything have a blue halo around it, for example, we use this little bit of code where from that CalX image, we use the get mask method to get the mask, and then you just loop over the mask plane dictionary and print the name of the mask and the color of each one. And you can actually set these colors. If you don't like the interpolated pixels to be green and you want them to be lavender, you can do that. And then yeah, and you can also, as I showed you, you can change. You can for example, hide all of the layers in this menu, this overlays tab, and then we can put them back. You can also change the color of them from within that tab. So I assume if I click that, yeah, there you go, and you can see then you can customize the color as much as you want. Oh right, I'm not showing a saturated star, so we're not seeing any interpolated pixels. Let's try changing the detected mask. If we instead want that to be, I don't know, magenta. Let's try it. There we go. So you have a lot of controls here to manually set aspects of the display here, but you can also do it from the notebook as we showed you. So I'm going to get rid of the mask plane for now, and just show the image. And then we're going to display some sources on top of the image. And so this is as we did in the previous notebook. We're going to go to the Butler and get this source catalog. So this is a source table that corresponds to detections of objects on this image. And you can see the length of that has about 2,500 sources in it. And we can see what that table looks like just by using this as AstroPy method that's associated to it. That makes an AstroPy table, which you can see, well, it's a little hard to see here since I'm, here we are. You can see a few of the columns. You see RA deck, and this actually is, if you go to the bottom here, you can see this table is extremely wide, and you can see these are all of the columns that are in that table. But we can save that for another time, exploring the table. That just shows you how you can see it. But now to plot those sources onto the image in that's displayed in Firefly, we're going to use this AFW display method that is called dot, that will plot a dot. This goes through or this plots an individual source, or marks an individual source on the display. So what we want to do is actually loop over them all. So we do this, embed the this display dot task within a loop here, so that we can execute it for all the sources and then display them all at once. And so now you can see all of the sources marked on the image. If we zoom in, you can see they're circled. And so what this is using, this is saying plot a circle. That's what the O here is. And then it's 20 pixel, I assume, I can't, looks like it's 20 pixel radius. And you can change the color if you like. I don't know what see if Orchid, I think that's a named color. Maybe not. Oh, there it is. Okay. So you can change the color to whatever color you prefer. We'll put it back to orange for now. Okay, I don't know why those didn't go away or didn't change. And then to clear them, there is a erase method associated with the AFW display. So if I want to get rid of those, boom, I just execute this erase. So we can put them back and confirm that orange came back. There we go. And then we can erase them if we want them cleared. Okay, and then the last thing we're going to do is, as we did in the other image display notebook, we're going to display a co-add image. So that'll be a deeper image. It's a stack of, some tens or maybe 100 individual visits. And so we're just going to select this data ID that is tracked 4431, patch 17, the I-band image. And then we fetch that image from the Butler. We use butler.get, and this is called deep coad calexp. That's calibrated exposure. So we'll retrieve that image using this, and we give it the data ID that we defined in the first line there. And then to get the measurements, we're going to get these two tables. One is the forced source, which is basically once the detection has been run, you force a measurement to happen at every position of a detection. And then this reference table just contains some more information about the objects that were detected from all of the bands, not just this I-band image. And so we're going to get those catalogs, and then we'll, in a minute, display those on the image. But okay, so now those have loaded. So now let's display that image in the Firefly display. And Eureka, I found a rich galaxy cluster in this image. Bet you didn't see that coming. Let's hide the mask planes so that we can see the images. Although actually that was interesting, because in this one you can see more of the mask plane set. You can see these cosmic rays in magenta color, which I don't think I noticed in the calyx. So that's showing illustrating another one of the mask planes. But we want to hide them so that we can overplot the sources on the display. And so in this case, we're just going to do a corner of the image here. So we're just going to do X and Y coordinates less than 1,000. So that should just be a lower corner here, just to make things go faster. And we're going to use this flag to select non-duplicate objects that have been deblended. You can read more about that. And then we're going to actually separately use a flag that identifies galaxies or point sources. So this is a flag that's in the source or in the tables that you can also read a little more about here. But basically it's a binary flag. It's set to 1 for extended sources, galaxies, and it's set to 0 for point sources like stars or quasars maybe. I modified this playing around with it yesterday. So here, let's go back to the original. This is what you should have. It'll say select. So we just extract the X and Y coordinates, and we'll note that the coad, the initial XY coordinates may not start at 0, 0. So we actually are going to extract that. And in fact, we'll just print that to the screen to see if that's the case for this image. Yeah, in fact, so the XY, the corner is actually at XY of 11,900, 7,900. And so you need to subtract that off of the XY coordinates that are in the table. And so now we just select the XY less than 1,000, and we apply these flags that we mentioned. So now we'll display the stars, the ones that come out from this is stellar flag.
Okay.
As orange. And so you can see those are objects that are, that are classified as stars. And then we can just display all of the extended sources, galaxies. These should be blue. Yeah, and so now you see you've done some, okay, I don't like my scaling here, but you've done some selection on the types of sources. So I just changed the stretch here with this stretch menu. So now maybe you can see a little better. So this orange circle here is circling at something that's classified as a star, and this kind of irregular looking thing next to it is classified as a galaxy. And so you could play around with selecting all different types of sources so that you can highlight whatever sources you are particularly interested in. And of course, you can do a variety of other things. This is just scratching the surface, but just wanted to show you different ways to display images. And if we have any questions, we can go to that. I don't know how we are on time. Yeah, we're already past the hour. So were there any questions?
There are some people reporting that their kernels are dying as they try to execute cells in a notebook.
I was wondering whether I noticed some people mentioned that they had started a small kernel, and my guess is that loading the coad image, it might be too large for a small kernel. So you probably need at least a medium or container. A medium.
Yeah, I think the recommendation is always to use medium if you're if you're doing image displays.
Yeah.
I've been discussing that with people offline here after I saw these messages starting. So yeah, that's the issue here.
Yeah, and I see Simona has a question about the star galaxy classification. And it's a simple classifier. I don't know if I think your question, how much can we trust it, is a very good one. That's something worthy of some exploration. That it's a very simple comparison of I think it's the C model, the model, galaxy model flux to the PSF flux. So that if if there's excess flux in the C model relative to the PSF, then it must be an extended source. And so it's based on that, but one could certainly derive a more effective star galaxy separation.
Yeah, and just to answer more directly, it can't be based on Gaia because this is a simulated data set.
Yes, also.
So you don't need even to do the unresolved resolved, or you know which one they are the
Yeah, but this is not based on truth. This is based. This is based on the data.
Thank
But
you.
wouldn't that be a valuable test, though? I mean, granted, Gaia is way too bright, but let's try to focus on the paint 10. Load up a Gaia image and run the sim algorithm. Have you guys done something like that?
No,
We
but
don't have a Gaia. I mean, it's not really directly comparable. I mean, we could use an HSC image.
How is it not?
That
And
would
then
be-
what
that would
about
be good.
QMAS? Same idea. See what I mean?
Yeah. No, the truth comparisons can be done in the simulation, and I think the team the desk team it has done and is doing those kinds of comparisons to see how well these classifications do. I mean, it's not the topic of this session, but those kinds of checks are definitely being done on the simulation side. And the
Okay.
truth data will be available in the science platform fairly soon. I think maybe a matter of another couple of weeks, I think, if I remember correctly. So you'll be able to do those kinds of tests yourself then as well.
And I have a notebook in our delegate contributions repository from DP0.1 that does exactly that. That I'm just waiting on the truth data for DP0.2 to update it and make it available.
So does that mean that we will be able to superpose catalogs to identify the stars, because they can
Yes.
contaminate the high ratio galaxies, right? So we will superpose this like in Aladdin. Or
Yes, you would be able
yeah,
to, at that
perfect.
point, you would be able to superpose the truth catalog on the on these images. Yes.
And external catalogs, or just the true simulation catalog?
Well, there are no external catalogs. that
Yeah,
are
but like
relevant.
you spoke about HCS images.
HS.
Those
Oh
are
no! So
real.
that was a response to a different suggestion that we try out that algorithm on real data. So
Okay. I was referring to that one,
That's
actually.
not going to be realistically doable in the DP0.2 environment.
Oh, okay.
Because the only data we have in that, the only image data we have in that environment is from the simulation.
Okay, thank you very much. That's very useful information,
Yeah.
actually, because I was I'm learning environments also for commissioning, and I guess we will have to work in another environment if we want to work on real data. Then.
Oh, there will be, yeah. I mean, this is the same science platform, but with different data. I mean, it will have the commissioning data visible in it as well.
Okay,
it's just a
fantastic.
different
Thank you so
instance
much.
of the same software. Yes.
Yeah, thank you so much.
Yeah. I see a question about the truth match. I think it probably would be simplest if we maybe Melissa, if we schedule a presentation on that once the truth tables are in the database. Is that possible?
Oh, that is the plan. Yeah. I
great.
mean, we have a bunch of tutorials that touch truth data that just, we can't update them yet because the truth data is not available. So once truth data will be released in a couple of weeks, everyone will get a notification that the truth data is available. It's going to take us a couple of weeks past that to actually update our tutorials and make sure that they are working with the new truth match catalogs. And then we will be able to start having tutorials in the delegate assemblies on those once they're updated. So everyone will be able to use the truth data though, like as soon as it's available, we'll be able to update the documentation. But yeah, let's leave everything truth data related later for later, and because we can't say much about it anyway right now. And let's set up our breakout rooms for the second hour. And also thanks Jeff for presenting. Thanks Gregory for being here. Thank you, Me, for also presenting. I know a bunch of people need to leave for the second hour, so we will say goodbye to those who are leaving. And everyone who wants to stay, stick around, do some breakouts.
Thank you very much. Bye-bye.
Yeah. I'm gonna share my screen. Let's set out some breakout rooms.